My name is Joshua Mugabi. I welcome you to topic 4, respiration. Our learning outcome B. Analyze the biochemical processes leading to ATP production in living organisms and how these processes are affected by physical activities and respiratory poisons, especially cyanide. Details of biochemistry are not required. We are going to look at a learning activity D. In groups, learners search for oxidative phosphorylation in the mitochondrion. The learners watch a video clip or role play the event of electron transport, focusing on the role of protein complexes, NADH, FADH2, and oxygen. Make a write-up and present. Oxidative phosphorylation and electron transport, item 1. We shall focus on the site and the meaning of oxidative phosphorylation. A student observes that most of ATP is in aerobic respiration is formed in the mitochondrion. Task: define oxidative phosphorylation. 2. State where oxidative phosphorylation occurs in the mitochondrion. Explain why the inner mitochondrial membrane is suitable for this process. Define oxidative phosphorylation. Oxidative phosphorylation is the synthesis of ATP from ADP and inorganic phosphate using energy released from the transfer of electrons through the electron transport chain to oxygen. State where oxidative phosphorylation occurs in the mitochondrion. It occurs in the inner mitochondrial membrane, the cristae. Explain why the inner mitochondrial membrane is suitable for this process. The inner membrane contains protein complexes of the electron transport chain. Cristae increases surface area for ATP production. The membrane is impermeable to protons, allowing formation of a proton gradient. Oxidative phosphorylation and electron transport, item 2. Focusing on the role of NADH and FADH2. During aerobic respiration, reduced coenzymes formed area are used in the mitochondrion. Task: state how NADH and FADH2 are formed. Explain their role in oxidative phosphorylation. State how NADH and FADH2 are formed. NADH is formed during glycolysis, the link reaction, and the Krebs cycle. FADH2 is formed during the Krebs cycle. Explain their role in oxidative phosphorylation. NADH and FADH2 donate electrons to the electron transport chain. They are oxidized back to NAD plus and FAD. Energy released drives the proton pumping across the inner membrane. Oxidative phosphorylation and electron transport item 3. Focusing on protein complexes in the electron transport chain. Learners study a diagram showing a series of protein complexes embedded in the inner mitochondrial membrane. Task: name the main protein complexes involved in electron transport. Describe the role of these complexes. Name the main protein complexes involved in electron transport. We have complex 1, which can be called NADH dehydrogenase. Then complex 2, which is also called FADH2 dehydrogenase. Then we have complex 3, which we call cytochrome complex. And then complex 4, which we can call cytochrome oxidase. Describe the role of these complexes. They transfer electrons stepwise along the chain. They release energy gradually. They pump protons from the matrix into the intermembrane space. That is complex 1, complex 3, and complex 4. Oxidative phosphorylation and electron transport, item 4. Focusing on the role of oxygen in electron transport. A cell deprived of oxygen stops producing ATP. Task: state the role of oxygen in the electron transport chain. Explain what happens when oxygen is absent. State the role of oxygen in the electron transport chain. Oxygen acts as a final electron acceptor. It combines with electrons and protons to form water. Explain what happens when oxygen is absent. Electrons accumulate in the chain. NADH and FADH2 cannot be oxidized. Proton gradient collapses. ATP synthesis stops. Oxidative phosphorylation and electron transport, item 5. ATP formation by chemiosynthesis. A proton gradient is established across the inner mitochondrial membrane. Task: explain how proton gradient is formed. Describe how ATP is synthesized using this gradient. Explain how the proton gradient is formed. Energy from electron transfer pumps protons into the intermembrane space. This creates a high proton concentration outside the matrix. Describe how ATP is synthesized using this gradient. Protons flow back into the matrix through ATP synthase. Energy released drives phosphorylation of ADP to ATP. This process is called chemiosmosis. Oxidative phosphorylation and electron transport, item 6. Focusing on the significance of oxidative phosphorylation. A student notes that most ATP comes from oxidative phosphorylation. Task: state the significance of oxidative phosphorylation. Give one consequence of damage to the inner mitochondrial membrane. State the significance of oxidative phosphorylation. Produces the majority of ATP in aerobic respiration. Supplies energy for cellular activities. Give one consequence of damage to the inner mitochondrial membrane. Reduced ATP production, cell energy deficiency, possible cell death. Marking emphasis. Correctly define oxidative phosphorylation. Describe electron transport in correct sequence. Explain the role of NADH and FADH2 protein complexes and oxygen. Link structure of mitochondrion to function. Use correct biological terminology. Summary. Oxidative phosphorylation occurs on the inner mitochondrial membrane and involves the transfer of electrons from NADH and FADH2 through protein complexes to oxygen. The energy released pumps proton across the membrane, and ATP is synthesized by chemiosmosis through ATP synthase, producing most of the ATP required by the cell. Thank you for participating in topic 4 respiration. Learning outcome B. Analyze the biochemical processes leading to ATP production in living organisms and how these processes are affected by physical activities and respiratory poisons like cyanide. Details of biochemistry are not required. We have been looking at learning activity D. In groups, learners search for oxidative phosphorylation in the mitochondrion. Learners watch a video clip or role-play the events of electron transport, focusing on the role of the protein complex, NADH, FADH2, and oxygen. Make a write-up and present. If this has been useful, visit me at Mugabi Joshua Biology on YouTube.